Good afternoon, everyone. Okay, so today we are going to discuss about lymphatic system, histology of lymphatic system. Now, in lymphatic system, there are lymphoid tissues, then lymphoid organs are also there. So this is the first time that you will be studying histology of any organs. Okay, till now, whatever you have studied, that was the general histology. From now onwards, we are entering into the systemic histology. Okay, where different organs will be there. We'll be discussing the microscopic structure of those organs. So today we will come to know the structure of this lymphatic organs, microscopic structure. Okay, so the concept is quite different. Okay, because earlier we have studied about the epithelial tissue, connective tissue, different types of tissues we have studied. Now there will be application of those tissues. Several times you will see that it is mentioned connective tissue, then different types of epithelial tissues will also be mentioned over here. So whatever we have discussed earlier, it will be a combination of all those things. Along with that, some new concept will also be there, which you have to understand. Okay. So we'll start with the lymphatic system. Now, before going to the microscopic structure of the lymphatic system, first of all, you must know what is the lymphatic system. Okay. So this lymphatic system is nothing but the you can say component of a drainage system, or you can say accessory to the venous system. Okay. Now in venous system, we know that the blood is draining back to the heart with the help of the veins. Okay. Now this lymphatic system is a accessory to the venous system. That means through that something is flowing. What is flowing? The lymph is flowing, and again it is going back to the venous system, and then from venous system it is reaching the heart. Okay. So that is the accessory, meaning of accessory to the venous system. Now, most of the tissue fluid, if you see, they are formed at arterial end of the capillaries. In the last class, if you remember, we have discussed about the blood vessels, right? Microscopic structure of blood vessel. Now, in the microscopic structure of blood vessels, we have discussed about the arteries, then arterioles, then capillaries. Okay, I have shown you the structure of capillaries. Now, what happens whenever the blood is flowing through the arterial end of the capillaries? At that time, most of the tissue fluids are formed, okay, and are absorbed back into the vessels by the venous end of the capillaries. So, arterioles, after that capillary bed, in that particular area, most of the tissue fluids are formed, and then it will be circulated or it will be distributed in the various tissues. And after that, again it is absorbed, or you can say again it is going back into the venous system, or you can say venous end of the capillary. From there it will go back to the venous system. We have discussed, I know you have not revised, okay, but we have discussed that thing in the last class. Now what happened? The remaining tissue fluid, now mostly they are draining back into the venous end of the capillaries. From there it is going to the venous system mostly, but some are left behind. Now those will enter inside the lymphatic channel. Okay. As you can see, the remaining tissue fluid are absorbed by the lymph capillaries. Now those lymph capillaries, they are present as a blind end in those tissue spaces. Okay. So from there, most of the fluid is going back into the venous system. Some are not able to go back to the venous system. They are absorbed by the capillaries, lymph capillaries. Okay. Now which begin blindly in the tissue spaces and is sent back into the blood. Now this lymphatic system is a closed system, okay, where some channels are there. After that, lymph vessels are there through which the lymph is flowing, and it is a one-way system. Okay, this is a one-way system. That means from lymph capillaries, then lymph channels, then lymph vessel, and ultimately it will drain into the venous system. And from there it will go back into the heart. This is how the lymph is flowing. Let me show you a diagram. I hope you can see this diagram. Now if you concentrate over here, then you can see a capillary bed is being formed. Okay. So this is the arterial end of the capillary, this is the venous end of the capillary, and this is the capillary bed, and you can see the tissue space is there. ओके तो क्या होता है, whenever the blood is flowing like this through the arteries, oxygenated blood carrying nutrients, oxygen, it is flowing like this, ठीक है. Most of the tissue fluids are formed over here. Then this tissue fluid will go and will supply the tissue spaces where tissues are there. After that, what will happen? Most of them, you can say approximately 90% of them, they are absorbed back by the venous end of the capillaries, and then through veins it is going back into the heart. ठीक है. But this small 10% which is remaining, what they will do? They are absorbed by these capillaries. Okay, this blind end of the capillaries. So ये जो capillaries है, these are lymphatic capillaries. तो उसका जो beginning होता है, that is present as a blind end in between the tissue spaces. So the fluid which is going back into the venous system, वो तो चला जाता है. जो remaining 10% रहता है, where they will go? They are absorbed by the lymph capillaries in the form of lymph. And after that, you can see they are flowing through the lymph vessel. Now, when they are flowing through the lymph vessel, ultimately they will drain into the venous system. And from this venous system, they will go into the blood. So this is one-way channel, as you can see. This is one-way channel by which the lymph is flowing. Now, when the lymph is flowing at that time, several structures are coming. For example, lymph capillaries are there, which are the smallest lymph vessels. Okay. Then large, medium-sized lymph vessels are there. Larger lymph vessels are there. Okay. Can anyone tell me which is the largest lymphatic vessel of our body? It is the thoracic duct. Okay, thoracic duct is the largest lymphatic vessel of our body. You will study this in thorax. Okay, next week or I think this week onwards thorax will start. So you will study this in thorax, the largest lymphatic vessel of our body, thoracic duct. Okay, so this is the concept of the lymphatics. Now when it is flowing like this, the flow is not that simple. Along with the lymphatic vessels, some organs are also there. Okay, now the question is why these organs are present? Because whenever the lymph is flowing, okay, it is having some important function. To fulfill those functions, these organs are present in between. Okay, so we have to understand first what are the functions of the lymph, then you will be understanding about the functions of those organs. Now here you can see the formation of the lymph. Whatever I have given you, that is a brief introduction that how lymph is produced and how it is entering into the lymph capillaries and how the lymph is flowing. Okay, but not in detail, just the introduction. Here you can see the lymph which I have shown you just now, where it is formed. Okay, now what are the constituents of the lymph? Now here you can see these are the constituents of the lymph. Okay, only 4% solids are there and 96% water is present. Okay, so organic substances are there and inorganic substances are also present, and some cellular contents are also there. For example, lymphocytes, monocytes, macrophages, plasma cells. These cellular components or cells are also present in the lymph. Okay? Details will be explained in physiology class. Next, the rate of flow. As you can see how the lymph is flowing, approximately 120 milliliter of lymph is flowing per hour into the blood. Okay, out of this 100 ml per hour is flowing through the thoracic duct. As I mentioned, thoracic duct is the largest lymphatic vessel of our body. Okay, next 20 milliliter per hour flows to the right lymphatic duct. That is the second largest lymphatic vessel of our body. Right lymphatic duct. Thoracic duct left side में रहता है, right lymphatic duct right side में रहता है. Okay, now there are some factors which are increasing the flow of the lymph: interstitial fluid pressure, blood capillary pressure, surface area of lymph capillaries, functional activities of the tissue, permeability of the lymph capillaries. These are some factors which will increase the lymph flow. Means at that time you will see that the flow is more. Okay, but in general, ideal condition it is 120 ml per hour. Next, what are the functions of the lymph, anyone? When the lymph is flowing through the lymphatic channels, then what do you think? What are different functions of the lymph?
Distribution of fluid throughout the body.
Yes, very good.
Interstitial fluid
Is done by the lymph. Now, lymph flow is responsible for maintaining the structural and functional integrity of the tissue, Then it serves as important route for intestinal fat absorption. तो whatever you are eating, अगर उसमें fat है तो उसका जो absorption होता है, after that the pathway which is followed, that is different. Okay, and for that the lymphatics are very important. I'll give you an example. When you will study small intestine, ठीक है, 3 parts of the small intestine का, duodenum, jejunum and ileum. Mostly absorption is taking place mostly in the duodenum. So in duodenum you will see that villi are present. Okay, so in villi, when you will study the microscopic structure of villi, at that time you will come to know that lacteals are present. These are nothing but the blind end of these lymphatic channels through which fat is absorbed. Okay, so at that time we will discuss how this is taking place, fat absorption. Then it plays important role in immunity by transport of lymphocytes. Now how this is taking place? Whenever this lymph is flowing through these different lymphoid organs or lymphoid tissue, at that time whenever it is required, some lymphocytes are also added through it. Okay, and it filters the lymph also. So that's why it provides the immunological support. So these are some important functions of the lymph. Next, if you see the lymphatic system in our body, so there are several components of the lymphatic systems. What are these components? First of all, lymph. Obviously, lymph will be there because it has to flow, and we have seen how the lymph is produced and the constituent of the lymph. Then once the lymph is produced, it has to flow. How it will flow? With the help of different lymphatic vessels. जो सबसे छोटा है, that is called as lymph capillaries. ठीक है, that is the lymph capillaries. Then larger one is the lymph vessels, and after that we are having lymphoid organs. Okay, so these are the constituents of lymphatic system. Now lymphoid tissues are also there. Okay, lymphatic tissue. So mainly 2 types of lymphatic tissues are there. What are these 2 types? Diffuse lymphoid tissue and dense lymphoid tissue. Okay, so based on whether capsule is present around the lymphoid tissue or not, it is divided into 2 types. Okay, now here you can see it is not enclosed by capsule, connective tissue capsule. That's why it is called as diffuse lymphoid tissue, and it is enclosed within capsule. That's why it is called as dense lymphoid tissue. Okay, 2 types are there. Here we are getting lymphocyte distributed in mucosa associated lymphoid tissue, also called as MALT. Okay, so when you will study the respiratory system, okay, in thorax we will study the respiratory system. Okay, in respiratory system you have to study about the trachea, you have to study about the bronchial tree. Okay, at that time this thing will be there, mucosa associated lymphoid tissue. Okay, at that time it is called as BALT, bronchus associated lymphoid tissue. Okay, next if you see the dense lymphoid tissue, then here we are getting the organs: lymph node, tonsil, thymus, spleen. Okay, these are the organs, mature lymphoid organs, and they are completely covered by connective tissue capsule. Okay, so this is the complete classification of the lymphoid tissue. So next about the lymph capillaries. Now 2 types of lymphatic vessels are present in our body: lymph capillaries and then lymph vessels. Now if you see the lymph capillaries, what are these lymph capillaries? They begin in the tissue as blind-ended sac. Okay, as a blind-ended sac. Now if you see the structure, first of all they are lined by endothelium, same like the blood vessel, the blood capillaries which are present. Okay, similar to the endothelium, blood capillaries, lymphatic capillaries are also lined by lymphatic endothelium. Okay, in case of lymphatic capillaries, these are more permeable as compared to blood capillaries. Okay, because of that only they permit to entry of absorbed larger lipid molecules through intestine. Permeability is more, that's why larger molecules can enter the lymphatic system. Okay, because the permeability is higher. Next, most of the tissues are having lymph capillaries, but there are some areas in our body where lymph capillaries are absent. Okay, for example, epidermis, cornea, cartilage, splenic pulp. bone marrow, choroid, internal layer, meninges, nervous tissue. These are the areas where lymph capillaries are absent. Okay, exception रहता है. Next, most of the lymph capillaries carry lymph towards the lymph node through afferent lymph vessel. Now this is very important because after that we are going to discuss about the lymphoid organs. Okay? So at that time we will come to know two types of lymph vessels are there. Lymph capillaries are afferent and efferent. Okay? तो afferent में क्या होता है? It will carry the lymph towards the lymph nodes, means towards the organ. It will carry the lymph. It is called as afferent capillaries. And efferent means they will carry the lymph away from the lymph nodes. That is the efferent capillaries. Two different types of capillaries are there. ठीक है? तो क्या होता है? Whenever the lymph is going towards the lymph node. Now जितना भी lymphoid organs होता है, among them majority are lymph nodes. जितना भी lymphoid organ होता है, for example, tonsil is there. Then spleen is there, thymus is there. Okay, so how many spleen is present? Only one. How many thymus is there? Only one. But if you see lymph nodes, plenty of lymph nodes are there. In each and every, almost every portions of our body, lymph nodes are present. Okay, so number of lymph nodes are more as compared to the other lymphatic organs. Okay, तो क्या होता है? In the pathway of the flow of lymph, some lymph nodes will be there. Now those lymph nodes, what will happen once the lymph is entering the lymph node, they will enter through this afferent lymphatic vessel, and after that the filtration of the lymph is taking place inside the lymph node. We'll discuss the structure today, don't worry. इसके बाद जो निकलता है filtered lymph, they will come out through the efferent lymph vessels. Okay, तो ये जो निकल के आता है, that is the filtered lymph. So at that time, if there is any pathogen or any unwanted substance, that will get filtered by that lymph node. And if required, it will add more amount of T lymphocytes in that particular lymph, which is coming out from the lymph node. Okay. Next, larger lymphatic vessel. So if you remember the layers of the blood vessel, how many layers are there? Last bench. Anyone from the last bench? How many layers are there in the any blood vessel? Name them. Tunica intima, not media. Tunica intima, then tunica media, and then adventitia. Outer one is the adventitia. So similarly, in case of larger lymphatic vessel, all those three layers are present. Okay. Now, lymph capillaries are the joint to form the lymph vessels. Now larger lymph vessels, thoracic duct, right lymphatic duct, jugular lymph trunk, subclavian trunk. Okay, these are different lymphatic vessels, larger lymphatic vessels of our body. Now if you see, large lymph vessels show tunica intima, media, and adventitia, same like the vein. Structure same रहता है, but there are some differences. Now what are those differences? Elastic fibers are present in all layers of the lymphatic vessels. This is first difference. Then circular smooth muscle cells are present in tunica media and adventitia. Okay, circular smooth muscle. And in thoracic duct, smooth muscles are arranged longitudinally. So these are some differences, or else mostly three layers are present and structure are similar than that of the veins. So veins का जो structure है, large vein, large lymphatic duct का जो structure है same रहता है. Only these three differences are there. But in case of larger lymphatic vessel, mostly valves are present. Okay, large vein में valves नहीं रहता है, medium size vein में valves रहता है. But in case of large lymphatic vessels, valves are present. So when you will study the thoracic duct, at that time you will study that valves are present inside the thoracic duct. Okay. Next, जो lymphoid organs है, तो just now I have shown you the list of lymphoid organs. उसमें भी दो टाइप का होता है, primary lymphoid organs and secondary lymphoid organ. Okay. Now primary lymphoid organ, they produce immature lymphocytes. Okay. For example, thymus and bone marrow. Here lymphocytes are present, but they are immature. That's why they are called as primary lymphoid organs. Secondary lymphoid organs, they provide exposure to the foreign antigen and convert immature lymphocytes to mature lymphocytes. Okay, so mostly spleen, lymph node, tonsil, appendix, pairs, patches, which are present in the intestine. Okay, so they are the examples of secondary lymphoid organs. So we will be discussing about both primary and secondary. Okay, the first structure which we have to discuss, that will be the spleen. Now before going to the spleen, in case of lymphatic system, I have mentioned about the lymphoid tissue also, where I have told you about two different types of lymphoid tissue. One is inside the capsule, or another one is outside the capsule. Okay. Now in that different types of cells are present. You cannot miss the cells. कोई भी सिस्टम है, ठीक है, उसमें ऑर्गन्स रहेगा, उसमें टिश्यू रहेगा. Now inside the tissue what will present? Cells will be there in the inside the tissue. Common sense. So similarly here you can see the list of cells which are present in the lymphatic system. ठीक है. Mainly three types of cells are there: T cells, B cells, and natural killer or non-T B cells. Okay. Now again T cells. These are the different types of cells. Now here we are not going in detail because these things you will study in physiology. Okay, the different types of cells present in lymphatic system. Just to give you an overview, that don't forget cells are also present. Okay, because when we will go to the organs, at that time you will see that organs are present inside the lymphatic system. Okay. So we'll start with the organs. The first one is the microscopic structure of spleen. Actually, I was planning to take this part during the time of abdomen. Okay, why? Because spleen is present in case of abdomen. Okay, but though it is there in the lymphatic system, so let's discuss it now only. Now in the microscopic structure, now before going to the microscopic structure of this lymphoid organs, please let me tell you one thing. Wait. What happens in case of lymphoid tissue, you will see that one similarity is there. What is that similarity? The lymphoid organ is enclosed in a connective tissue capsule. All the lymphoid organs, you will see that they are enclosed in a connective tissue capsule. Okay? Next, from that connective tissue capsule, the septa is arising, or you can say trabeculae is arising, and that is going deep into the organ or deep into the structure, and then it is dividing it into different lobules. Okay? Now, if you consider one particular lobule, then you will be getting two things. Okay? Now, what are those two things? Any guess? One is cortex and another one is medulla. Cortex is the outermost area, medulla is the innermost part. Okay, so this concept of cortex and medulla is present in all the lymphoid organs. Almost all the lymphoid organs, not all. In case of tonsil, some differences are there. Okay, but mostly similar structure will be there. तो पहले क्या आता है? Connective tissue capsule. ठीक है? From the capsule, septa is going inside the gland or inside the structure, divided into small lobules. If you take one lobule, then the outer part is the cortex, inner part is the medulla. Okay? Now, in the cortex, what happens? Mostly you will see that the appearance of the cortex is darker in nature. When you will prepare a H&E stain slide, in most of the lymphoid organs, you will see that जो cortex होता है, that is darkly stained. Okay, basophilic होता है, darkly stained होता है. I will show you the diagrams also. In comparison to that, the medulla is lightly stained. So these two differences are more or less common for most of the lymphoid organs. Okay, so these are some similarities which you will find in each and every lymphoid organs. Next, what you will see next, whatever things we will discuss, so that varies from organ to organ. जैसे spleen में अलग होगा, thymus में अलग होगा, lymph node में अलग होगा. Okay, but these points, whatever I have mentioned, those are similar for all the organs. Next, if you remember, I have told you that जो lymphoid organs होता है, उसका जो framework है, that is made up of reticular fibers. जो लिम्फॉइड ऑर्गन्स होता है, जितना भी लिम्फॉइड ऑर्गन्स होता है उसका जो कनेक्टिव टिश्यू फ्रेमवर्क होता है, दैट इज मेड अप ऑफ रेटिकुलर फाइबर्स. So here also you will see that several times I will be mentioning about the reticular fibers. Okay. Now, if you see the microscopic structure of spleen, so as you know, spleen is the largest lymphoid organ of our body. Okay. What is the function of the spleen? It filters the blood and it removes the old RBC. Okay. That's why it is also called as graveyard of the RBC. Okay. Now since it is in the bloodstream, so it filters the blood-borne antigens and microorganisms. So though it is connected to the bloodstream, that means blood is continuously entering the spleen, getting filtered. So blood-borne antigens, it will also enter the spleen. Okay, and that has to get filtered. Along with the older RBC, those things are also filtered. Okay. Now, histologically, when you will see the structure of spleen, three things will be there. First of all, you will be getting the capsule. Okay? Then you will be getting the trabeculae, as I have mentioned, or in layman term you can say it as partitions. जो trabeculae है, in layman term you can call them as partitions. ठीक है? For example, suppose this is the roof. From the roof I have given so many partitions, I have drawn or I have put so many partitions. So then what will happen? This glass will be divided into small compartments. ठीक है? जैसे मान लो यहाँ पे एक पिलर है, तो I have given one partition over here, वहाँ पे एक पिलर है, again I have given one partition, तो क्या होगा? Classroom will be divided into small compartment, right? तो similarly जो spleen है उसमें कैप्सूल रहता है, तो कैप्सूल से ऐसे पार्टीशंस नीचे कनेक्टिव टिश्यू और ट्रेबेकुले, दे आर गोइंग डीप इनटू द ग्लैंड. आफ्टर दैट इट इज डिवाइडिंग इनटू स्मॉल लोब्यूल्स. ओके, so that is the capsule trabeculae, and inside the trabeculae, once you will see inside the spleen, at that time you will notice that two things are visible, that is the white pulp and red pulp. And you have to know what is white pulp, red pulp. You have to know the differences between white pulp and red pulp. Okay. Now this is the chart which will help you to understand the microscopic structure of the spleen. So first of all, capsule and trabeculae is there, which is covered by dense connective tissue. In here, one important cells are present, it is the myofibroblast cells, and after the trabeculae, which are arising from the capsule, okay, enters deep into the parenchyma of the gland. Okay. Now in the parenchyma, two things are there, that is the white pulp and red pulp. Two things are there, white pulp and red pulp. So we have to understand the concept of white pulp and red pulp, and then one marginal zone is there. Now if you see the white pulp and red pulp, okay, first of all, under H&E stain slide, when you will see the white pulp, it is darkly stained. Okay, first of all it will darkly stain, and what is the color? Basophilic. Basophilic है तो कलर क्या होगा? Appearance blue. B for B. Basophilic B, blue is also B. ठीक है? तो like this याद ऐसे करके याद रखो. तो basophilic appearance होगा मतलब blue color appearance, और in case of H&E stain slide purple appearance और dark violet appearance होगा. ठीक है? So that is the white pulp, and in that you will see a central arteriole is present. Now that central arteriole is mostly eccentric in nature, not concentric, eccentric in nature. Center में नहीं रहता है, थोड़ा सा towards the periphery रहेगा. ठीक है? Now, so this is the white pulp. Next, in case of the red pulp, we will see it is lightly stained and eosinophilic, means थोड़ा pinkish appearance होगा. Okay, so from there you can differentiate. This is the identifying features or differentiating features by which you can differentiate between the white pulp and red pulp. Now if you go more detailed inside the white pulp, then you will see that two things are present. Okay, now that is periarterial lymphatic sheath, which is made up of T cell, splenic nodules or Malpighian corpuscles, which are made up of B cells. तो इसमें क्या होता है, ये जो सेंट्रल आर्टेरियोल्स है, नाउ दिस सेंट्रल आर्टेरियोल्स आर सराउंडेड बाय T lymphocytes, then it is called as periarterial lymphatic sheath. If it is surrounded by B cells, okay, B lymphocytes, then it is called as splenic nodules or Malpighian corpuscles. Now the question is, when we are getting B cells and when we are getting T cells. In general, we are getting T cells, but if it is exposed to any pathogen, at that time B cell will proliferate. The number of B cells will get higher. At that time, we will be getting the Malpighian corpuscles, and then we will be getting the germinal center along with the eccentric arteriole. That indicates that body has been exposed to any type of antigen. Okay. Now, if you concentrate over here in the red pulp, as I said, first of all, it is lightly stained and it is eosinophilic in nature. Okay. Now, here also two things are present, that is cords of Billroth and splenic sinusoids. Now, what is this cords of Billroth? Now, here different types of cells are present. Okay, starting from RBC, lymphocyte, macrophages, plasma cells. Okay. And this red pulp, if you see, mainly the red pulp is in the form of a sponge. I hope you have seen sponge. So, जो red pulp होता है, it is in the form of sponge, where small spaces are there, and those spaces are pervaded, or you can say those spaces are occupied by these different types of cells, and those cells are irregularly arranged and anastomosing with each other. Okay. And thus they are forming the cords, and that is called as cords of Billroth. Next, what happens? Splenic sinusoids are there. Now, in between the cords of Billroth, small spaces are there, which are lined by endothelium, and that is called as splenic sinusoids. Okay. Now, in the splenic sinusoids, तो क्या होता है, जो cords of Billroth में क्या रहता है, different types of cells are there. And those cells are irregularly arranged, and उसके बीच में जो spaces होता है, tiny spaces, those are called as splenic sinusoids. In that you will see endothelial cells are present, but here the endothelial cells are not like normal endothelial cell. What is the which type of epithelium is the endothelium? Simple squamous epithelium. Now here the endothelial cells are elongated and they are banana shaped, and that's why they are called as stave cell. Okay. Now whenever the blood is passing through that sinusoids, okay, they can increase or decrease the lumen of the sinusoids. As a result, what will happen when older RBCs are passing through that sinusoids, okay, they will not allow the older RBC to pass through it. Thus they are filtering it. Okay, so that is the function of the stave cells, STAVE, stave cells, in case of splenic sinusoids. Okay. Next, a marginal zone is there, which lies where white pulp and red pulp comes in contact with each other. So suppose white pulp is surrounded by red pulp, okay, that is called as marginal zone. Now in marginal zone, antigen presenting cells are present. Okay. Now those antigen presenting cells will carry the pathogen, okay, and it will expose them to B lymphocytes, which will produce antibodies against them. That is called as marginal zone. Okay. So that is the structure of spleen. Now let me show you the diagram. Now first you have to see this one. Now I am not going in detail about the blood supply of spleen. Blood supply of spleen is a bit controversial. Two different theories are there, one open theory and one closed theory. Okay. Now that's why you can see it is written as open circulation as well as closed circulation. Once the blood is inside the spleen, at that time how the circulation is taking place, till now research is going on and it is controversial. Some says that it is open circulation, some say it is closed circulation. Now we will not discuss this because it will make it very complicated. Okay, this will keep it when we will start the abdomen. In abdomen, spleen will be discussed. At that time we will see all those things. Now for the time being, just keep that in mind that दो type का circulation रहता है in the spleen. Now can you see the capsule? Can you see the capsule? So this is the connective tissue capsule and this is the trabeculae. This is the trabeculae. ठीक है? तो क्या होता है? Through trabeculae, trabecular artery is entering inside the substance of the spleen. Okay. After that, this is called a central artery. Now, when the central artery is surrounded by T lymphocytes. Okay, you can see it is surrounded by T lymphocytes. This is how the white pulp is formed. Next, here you can see the periarterial lymphatic sheath is also there. That means this is the central arteriole and surrounded by it is the T lymphocyte. Okay. Next, what happens in some cases, instead of T lymphocytes, B lymphocytes proliferate, and a germinal center is also present. So at that time, what will happen? It will push the arteriole. Okay, and when you will take the section, it will become eccentric in nature. Center में नहीं रहेगा. Okay. So this is one diagram which is showing the white pulp. Okay, and this particular part is the red pulp. Another diagram is there. Wait. This is a diagram, important diagram showing the microscopic structure of spleen. तो इसमें क्या दिखाई दे रहा है? First of all, you can see this is the capsule. Okay, connective tissue capsule. Then here you can see the trabeculae which is extending towards the substance of the spleen. Okay. Now, as I have mentioned in histology or microscopic structure of spleen, first of all you have to see the capsule, then trabeculae which are coming from the capsule. After that, what you have to see? White pulp and red pulp. What is the appearance of the white pulp? Basophilic in nature, darkly stained basophilic in nature. तो यहाँ पे दिखाई दे रहा है डार्कली स्टेन बेसोफिलिक. ये जो राउंड राउंड दिखाई दे रहा है, ये देखो, ये जो राउंड राउंड दिखाई दे रहा है, these are white pulp. Okay, what is the appearance? Basophilic in nature, darkly stained. ठीक है. Now, can you see the arteriole? ये देखो, this is where is the position? Eccentric in nature, not concentric. ठीक है. Now, in some cases you can see germinal center is present. In some cases germinal centers are present, in some cases it is absent. ठीक है. तो ये देखो, this is the appearance of germinal center. तो germinal center actually होता क्या है? When it is exposed to pathogen, at that time immature cells are forming. Okay, so that it has to provide immunity or immunological support. So that's why there will be a center zone where it will be lightly stained. Why it is lightly stained? Because immature cells are present, and it will be surrounded by a darkly stained area where mature cells are present. That is the germinal center. Okay, so here you can see this is the germinal center, and see the position of the artery which is eccentric in nature. So this is the white pulp. Okay, next, so this is the white pulp. Okay, after that, see this is the red pulp. Okay, now in red pulp what you have to see? क्या देखना है red pulp में? As I have mentioned earlier, first of all, what is the appearance? It is eosinophilic. Okay, it is eosinophilic. Then two things are present. It is cords of Billroth and splenic sinusoids. Cords of Billroth mean, can you see these small dots? These are nothing but the cords of Billroth. मतलब यहाँ पे different types of cells are present, and those cells are irregularly arranged. They are branching and anastomosing with each other to form the cords. In between the cords, I hope you can see small white spaces can be seen. Those are nothing but the splenic sinusoids. Now sinusoids are extremely narrower that you cannot see under low magnification. It can be seen but not clearly. High magnification is required in order to see the splenic sinusoids. ठीक है. Splenic sinusoids में क्या रहता है? Endothelium रहता है but modified. Banana shaped. Stave cells are there. Okay, उसका main function क्या है? It controls the lumen of the sinusoid, so that whenever blood will flow, it can filter whatever unwanted materials are present in the blood which is passing through the spleen. ठीक है. So that is the structure of spleen. So please keep that in mind. In case of spleen, you have to write about the capsule, you have to write about the trabeculae, you have to write about the white pulp, and you have to write about the red pulp also. Okay. Now this is a schematic diagram. Now this is a low magnification image of spleen. ठीक है. तो इसमें क्या दिखाई दे रहा है, ये देखो पहले कैप्सूल दिखाई दे रहा है, ठीक है. देन हियर यू कैन सी द रेड पल्प, here you can see the red pulp where splenic cords can be seen. Next, this is the white pulp, this is also a white pulp, okay, and here you can see the eccentric position of the arteriole, ठीक है. So उतना देखना है, आइडेंटिफाई करना है, then you can say that this is a slide of spleen. Okay, so that's all about the spleen. Next is the lymph node. Next structure is the lymph node. Okay, so majority of the lymphoid organs are nothing but the lymph nodes. Okay, because in the upper limb you have studied about the axillary group of lymph nodes. ठीक है, do you remember the axillary group of lymph nodes? Five groups of axillary lymph nodes are there, and they are draining which part of the upper limb?
Breast.
Not only breast, it is draining different areas where upper limb is also included. ठीक है, तो अगर if someone is asking that axillary lymph nodes, I know it is 75% of the breast is drained by axillary lymph node, doesn't mean that only breast, other areas are also there. ठीक है. Now that day while mentioning about the lymphatic drainage, I have told you what is the meaning of this lymphatic drainage. You have studied that axillary lymph nodes are there and it is draining lymph from this area. इसका मतलब क्या है? What is the meaning of this? For example, 75% of the lymph from mammary gland or breast is drained by axillary group of lymph nodes. इसका मतलब क्या है? What is the meaning of this? Then what we have discussed so far? That is the concept that you have to understand, that you have to understood. See, in the beginning I have told you जो lymph है, production होता है lymph का, after that how it is flowing. First of all, it is absorbed by the lymph capillaries, which are nothing but the dilated blind end which are present in the tissue spaces. ठीक है, from there lymph is flowing. Now when the lymph is flowing, at that time what happens? In between the lymph vessels, different lymphoid organs will come. Okay, now among them, which is the abundant lymph organs? These are nothing but the lymph nodes. ठीक है, तो and in lymph nodes, as I have mentioned, how the lymph is coming through afferent lymphatic vessels and how it is going out from efferent lymphatic vessel. ठीक है, तो suppose mammary gland है, around the mammary gland the lymph nodes which are present. तो from this mammary gland the lymph which will come out, they has to pass through these lymph nodes. Okay, they will pass through these lymph nodes. Now when they are passing through these lymph nodes, it used to get filtered. Sometimes what happen, whenever they are passing through this lymph nodes, suppose a person is suffering from CA breast. Suppose when this lymph is passing through these lymph nodes, through afferent it will enter the lymph node, and after filtration it will come out through the efferent. ठीक है, तो suppose if a cancer cell is there, it will go into the lymphatic system, it will go into the lymph node. ठीक है, after that what will happen? It get filtered. Once it get filtered, the lymph will come out. Now those cancer cells used to be present over there inside the lymph node, and as a result the lymph node will get enlarged. Okay, nodule type formation होगा, lymph node used to get enlarged. In the similar way, if any infectious material is there, any pathogen is there, it can go and it can infect the lymph node also. Okay, so this is the meaning of the lymphatic drainage. Means ये जो particular area है, वहाँ से जो lymph निकलेगा, that has to pass through these lymph nodes. Okay, so we have to study the structure of lymph node. So if you see the lymph node, what is the shape? It is kidney shaped. I hope you have seen the diagram of kidney. ठीक है, तो जो lymph node होता है kidney shaped होता है, which filters the lymph. तो first of all it is having a convex surface and the concave area. तो kidney shaped एक convex रहेगा और एक concave area रहेगा. Through convex surface the lymph will enter, and through concave area it will come out from the lymph node. Okay. See, it enters through afferent lymphatic vessels and it comes out through efferent lymphatic vessel. जहाँ से निकल के आता है, it is called as the hilum of the lymph node. Okay, that is called a hilum of the lymph node. Now, through this area, the blood vessels also enters the lymph node. Here is the diagram. Okay, here you can see the diagram. So, can you see the kidney shaped area? ये देखो. So, this is convex area and this is the concave area. In the concave area, you are getting the hilum. ठीक है? Now, just see how the lymph is entering inside the lymph node through afferent lymphatic vessels. ठीक है? After that, there are different areas of the lymph node that we will discuss, and then how it is coming out. It is coming out through efferent lymphatic vessel. ठीक है? Now, this particular area, ये देखो, यहाँ से जो efferent lymphatic vessel निकल के जा रहा है, this is called as hilum. Hilum of the lymph node, and through this hilum of the lymph node only you can see the lymphatic vessels. Okay, that means lymphatic artery and vein which is supplying the lymph node, it is also entering inside the lymph node. Now we have to understand the microscopic structure. Okay, what is present over here, what is present over here, all around. Now this is the chart which will help you to understand about the microscopic structure of lymph node. Okay, story will remain same. Okay, which story? In the beginning I have mentioned one story about the lymphatic organ. पहले क्या रहेगा? नहीं, कैप्सूल. पहले कैप्सूल रहेगा, ठीक है? हाँ, इसके बाद ट्रेबेकुलर रहेगा, then cortex रहेगा, then medulla रहेगा. देखो, same thing is repeated. So first we are getting the capsule which is made up of dense connective tissue. Okay, from here trabeculae will be there which will go deep into the gland. Now next in the parenchyma two parts are there, cortex and medulla. So how to differentiate between cortex and medulla? Based on the staining. ठीक है, based on the staining. जो cortex है that is darkly stained, medulla lightly stained होता है. So see, cortex it is darkly stained, medulla it is lightly stained. Okay. Reticular meshwork as I have mentioned earlier, जो lymphatic organs होता है उसका जो framework होता है that is made up of reticular fibers. Don't forget that. Okay. तो supportive network जो होता है that is made up of reticular cells. Then we are getting reticular fibers, and along with that dendritic cells are also present. Okay. Dendritic cells are antigen presenting cells. Now, two things are there which you have to notice. That is, in case of parenchyma, we are getting cortex and medulla. Okay. In case of cortex, two different types of areas are there, and then we are getting a network of sinuses. Okay. These two things you have to follow in case of lymph node. Very important. Okay. And this will help you to identify the slide under microscope. Okay. So I am coming into the sinuses. Before that, let's discuss about the cortex. So first of all, cortex is darkly stained. ठीक है? Now, जो cortex होता है, two different areas are there of the cortex. Okay, this is the outer cortex, or also called as nodular cortex. Okay. Now in the nodular cortex, what happens? Just below the capsule, जो capsule है, just below the capsule, जो cortex का area है, it is called as outer cortex, or also called as nodular cortex. Now why it is called as nodular cortex? Because you will see the lymphatic nodules. Okay, that is aggregation of lymphatic tissue or aggregation of lymphocytes. Dark appearance is there, and along with that germinal centers are also present. Okay, over here the lymphatic nodules are there, germinal center is present. Okay, darker area is there, it is called dark corona, and it contains B cells and plasma cells predominantly. So these are the important identifying feature. Next, if you see the cortex which is in between the cortex and the medulla, that is called as paracortex, or also called as thymus dependent zone. Okay, here what happens, you will not see these lymphatic nodules. Instead of this, you will see only lymphocytes. Which type of lymphocytes? Only T lymphocytes are there.
तो जो नोड्यूलर कॉर्टेक्स है, मेडुला है, उसके बीच में ए नैरो जोन इज प्रेजेंट, दैट इज कॉल्ड एज पेराकॉर्टेक्स. ठीक है. Now in that paracortex, those nodules, germinal centers, all those things are not there. Instead of that, aggregation of T lymphocytes are present. That's why it is called as thymus dependent zone. Okay. Next, if you come to the medulla, it is lightly stained. Cords of B lymphocytes are there. Means B lymphocytes are more in case of medulla. Less number of B lymphocytes are present. That's why they are lightly stained. And cells are widely separated from each other, and in between the cells we are getting sinuses. ठीक है. Now if you come to the sinuses, in case of lymph node, 3 different types of sinuses are there. Okay, 3 different types of sinuses. A well defined sinuses, network of sinuses are present in the lymph node. First let's see about the sinuses. तो पहला जो sinus आता है, this is the subcapsular sinus, that is the important identifying feature of the lymph node. जो कैप्सूल होता है, ठीक, कैप्सूल के नीचे एक स्पेस दिखाई देगा. You will see a small space which is situated just below or deep to the capsule, that is called as subcapsular sinus. ठीक है, beneath the capsule. Next is the trabecular sinus. Adjacent to the trabeculae a sinus is present. तो कैप्सूल के बाद क्या आता है, ट्रेबेकुले आता है. तो adjacent to the trabeculae again sinus is present, that is called as trabecular sinus. And in the medulla, the sinus which is present, that is called as medullary sinuses. Okay, like this 3 different types of sinuses are there. Now please keep that in mind. These sinuses are very important. Why these sinuses are important? Once the lymph is entering into the lymph node, they has to flow those sinuses, and ultimately when they will reach the medullary sinus, they are drained by the efferent vessels. Please keep that in mind. जो lymph है, let me show you the diagram again. हाँ, here you can see. तो ये देखो, this is the efferent lymphatics, this is the efferent lymphatic vessel through which the lymph is entering. Once the lymph will enter, ये कैप्सूल है, ठीक, इसके नीचे यहाँ पे एक साइनस है, दिस इज कॉल्ड एज कैप्सुलर साइनस. ठीक है. इसके बाद ये जो ट्रेबेकुले है, एडजेसेंट द ट्रेबेकुले द साइनसेस आर प्रेजेंट, दोस आर कॉल्ड एज ट्रेबेकुलर साइनस. दिस आर द पाथवे थ्रू व्हिच lymph is flowing inside the lymph node. After that they will reach the medullary sinuses. ये देखो मेडुला में साइनसेस है, and then they come out through the efferent lymphatics. That's why these sinuses are very important. Now let me show you the diagram. So here you can see the diagram. This is the microscopic structure, low magnification image of lymph node. ठीक है. तो पहले क्या देखना है? First what you have to see? Capsule. First you have to see the connective tissue capsule. Can you identify the capsule? हाँ, ये देखो, ये capsule दिखाई दे रहा है. ठीक है. Now deep to the capsule, can you see a small space is there? This is called as subcapsular sinus. ठीक है. After that you can see this is the cortex. Now cortex में 2 एरिया होता है, the outer cortex and the paracortex. तो here you can see this is the outer cortex. ठीक है. ये जो पूरा एरिया है, this is outer cortex, this is outer cortex, this is outer cortex. ठीक है. And this is the trabeculae. ये देखो, this is the trabeculae. तो कैप्सूल से ट्रेबेकुले अंदर आ रहा है. As you can see, this is the trabeculae. Now this entire part is the cortex. Now what is present in the 2 areas are there in the cortex. One is the outer cortex, another one is the paracortex. In outer cortex what you will get? Lymphatic nodules. ये देखो, these are the lymphatic nodules. These are the lymphatic nodules. ठीक है. Now here you can see the germinal center. You can see the germinal center. ठीक है. In all the case germinal centers are not present. In some cases germinal centers are present. It depends on the whether it is exposed to pathogen or not. ठीक है. Now next, can you see this darker area? ये जो dark area दिखाई दे रहा है, this is called as deep cortex or paracortex. क्या हुआ? हाँ, तो जाओ बाहर जाके रेस्ट लो. Why you are sleeping inside the classroom? जाओ, go and take rest. No, go and take rest, no problem. जाओ. So this particular area, this is called deep cortex or also called as paracortex. Okay, इसमें क्या रहता है? What is present over here? T lymphocytes. Okay, here T lymphocytes are more, that's why it is called as thymus dependent zone. Okay, next if you come more deep, then यहाँ पे क्या मिलेगा, this entire part is medulla. So you can see the difference, staining differences. Medulla is lightly stained, this is darkly stained. Why? medulla में B lymphocyte predominant होता है, जो मेडुला है उसमें B lymphocytes predominant है, but the number of B lymphocytes are less. Okay, and in between the B lymphocytes, okay, you can see small spaces are there, and these are called as medullary sinuses. Okay, now what are the three different sinuses we have seen? First is the subcapsular sinus, then you can see the trabecular sinus, and then the medullary sinuses. Okay, इसका significance क्या है? The lymph is flowing through those sinuses. Okay, once it reaches the medullary sinus, then it will come out through the efferent vessels. Another image is there. So this is high magnification image. Okay, this is low magnification image. Previous one was schematic diagram. ठीक है, जो इससे पहले दिखाया था वो schematic diagram था, drawable diagram. Now this is the actual image. This is high magnification, this is low magnification, this is average. Okay, तो यहाँ पे क्या दिखाई दे रहा है? पहले देखो कैप्सूल दिखाई दे रहा है, इसके नीचे देखो सब कैप्सुलर साइनस दिखाई दे रहा है, ये ट्रेबेकुलर दिखाई दे रहा है. ठीक है, नेक्स्ट ये देखो, दिस इज द आउटर कॉर्टेक्स वेयर नोड्यूल्स कैन बी सीन. ये देखो यहाँ पे एक नोड्यूल दिखाई दे रहा है, यहाँ पे नोड्यूल दिखाई दे रहा है. ठीक है, नाउ इफ यू कम डीप, ये देखो जर्माइनल सेंटर भी दिखाई दे रहा है. ओके, नेक्स्ट in this particular area you will be getting the paracortex. ठीक है, यहाँ पे paracortex दिखाई देगा, जो paracortex दिखाई देगा वहाँ पे nodules will be absent. Next, if you come more deeper, you can see this is the medulla, lightly stained, as you can see the difference, staining difference, and so many spaces will be seen, small spaces. Those are medullary sinuses. ठीक है, so that's all about the lymph node. Okay. Next is the thymus. Thymus is a complicated structure. It's not that simple. Till now whatever we have discussed उतना simple नहीं है thymus का. Functions भी अलग है. Hormones are also secreted from the thymus. Okay, and along with that, it is a primary lymphoid organ. Till now whatever we have discussed, those were secondary lymphoid. Now thymus is a primary lymphoid organ. And because of the primary lymphoid organ, a barrier is present. Okay, that is called blood-thymic barrier. Thymus में जो T lymphocytes रहता है, those are not allowed, the antigens are not allowed to get exposed to those lymphocytes because they are immature. ठीक है, तो उसके लिए barrier देना होगा. Because you can't go and tell the antigens that don't go and expose them. Who will do that? For that a barrier is there, and you have to know the structure of the barrier. So that barrier is protecting it to get exposed by the antigen. Okay, that's why the structure थोड़ा सा complicated हो जाता है. And there is a group of epithelio-reticular cells, specialized epithelio-reticular cells. In case of thymus, 6 different types of cells are there. 3 in the medulla, 3 in the cortex, having specific functions. Okay, and along with that, they secretes variety of hormones also, which helps in the maturation of the T lymphocytes mostly. Okay, so let's start with the last one, that is the histology of thymus. Okay, तो क्या होता है exactly in case of thymus? First of all, thymus and bone marrow are the primary lymphoid organs. Don't forget that. ठीक है. Now in case of thymus, 2 lobes are there. That's why it is called bilobed structure. Okay, and it is present in the anterior part of the superior mediastinum. You'll be studying about the mediastinum soon. Okay, at that time you'll come to know that thymus is present. Now embryologically it develops from third pharyngeal pouch, and mesoderm contributes to the thymocytes. That is the development of the thymus. Now at birth, if you see the weight is 10 to 15 gram. Then at puberty the weight increases 30 to 40. Okay, and after that subsequently it reduces. Okay, and subsequently the organ is replaced by fat. Okay, so once the person is adult, at that time, if you, for example, in the cadavers, if you dissect now, you will not get the thymus. Okay, because it is replaced by fat. Now the thymus believed to produce the T lymphocyte throughout the life, but it is doubtable. It is having doubt, means question is still there whether it is producing or not T lymphocyte throughout the life. Now it is having rich blood supply and do not receive any lymph vessel but gives efferent vessel. This is the peculiarity in case of thymus. ठीक है, कोई भी efferent नहीं है. Why efferent is not there? Because it is primary. अगर आएगा तो expose हो जाएगा. हाँ, it should not. That's why. Next, here you can see the structure of the thymus. ठीक है, देखने में easy लगता है. As you will go inside, it will be difficult. So first of all, same thing is repeating. Capsule is there, covered by connective tissue capsule. Now here there is a catch. ठीक है. Now what is that? Septa is there which divides the thymus into incomplete lobules. ठीक है, तो जो lobules divide होता है, that is incomplete in nature. Next, if you take the parenchyma, here two things are there, same like the lymph node. Cortex is there and medulla is there. Here also you can see cortex is darkly stained, medulla is lightly stained. Same thing is present over here also. But in case of medulla, you will see an important structure is present, that is called as Husserl's corpuscle. Okay. In case of medulla, you will see a specialized structure present. In case of thymus, that is called as Husserl's corpuscle. Okay. That is an important identifying feature of thymus. Now what is this? It is a hyaline material surrounded concentrically by type 6 epithelio-reticular cell. I have mentioned that 6 different types of epithelio-reticular cells are present. So a hyaline mass is there. That means you will see a dark pink color structure surrounded by a group of cells, and those are called as epithelio-reticular cells, which are present concentrically. Okay. Next, epithelio-reticular cells, 6 types are there. Okay. And among them, the most important function of these cells are they are forming the blood-thymic barrier. Why this barrier is formed? Yes. See, isolate the T lymphocytes from the connective tissue, mainly from the blood, so that it can mature. Agar mature nahi hoga, then it cannot provide necessary protection. It cannot produce antibodies. That's why maturation is required. For that, this barrier is formed. Along with that, you can see production of mature T lymphocyte. This is one important function. Secretion of thymosin and thymopoietin. These are two important hormones which are produced by the thymus gland, and that is purely responsible for maturation of these T lymphocytes. Now, here you can see one thing. यहाँ पे क्या होता है? Another important feature is there. First of all, I hope you can see the connective tissue capsule. Then the trabeculae which are entering inside the gland, it is dividing it into incomplete lobule. First point. Second point, if you take one incomplete lobule, then you will see that outer part is cortex, inner part is medulla. ठीक है? Now, if you take two such structures, okay, at that time you will see जो medulla है of one lobule is continuing with the medulla of the another lobule. Okay, तो यहाँ पे you can see ये जो suppose this is the trabeculae, suppose this is the trabeculae, it is dividing it in incompletely. So this is the cortex, this is the medulla. तो क्या होगा? ये medulla जो है will continue with the medulla of this one, adjacent incomplete lobule. Suppose एक trabeculae है, दो lobule हो रहा है, तो दोनों lobule का cortex रहेगा, medulla रहेगा. तो दोनों जो medulla है lobule का, both are continue with each other. That is the specialty in case of thymus. Okay. Let's discuss this in detail, or else there will be some doubts. Okay, first of all, if you see the cortex, it is peripheral, darker, basophilic. In case of thymus, it contains closely packed T lymphocytes. Okay, closely packed T lymphocytes are present. Always keep that in mind. Thymus में कोई different types of lymphocytes are not there. Only T lymphocytes are present. Okay, and it is supported by a meshwork of epithelio-reticular cells. Very important. Okay. Now we will see the functions of these epithelio-reticular cells, and mostly they are derived from third pharyngeal pouch. In case of cortex, macrophages are also there, which helps in phagocytic activities. Next, in the medulla, cortex and medulla is very simple. In case of thymus, easily it can be identified. So it is a lighter zone of the thymus. That means lightly stained area of the thymus. Now, medulla of each thymic lobule is continuous with the medulla of adjacent lobule, as I have mentioned. Okay, continuation rahega because lobules are incomplete in nature. Next, medulla is having less density of thymocytes. That's why it is lightly stained. But it contains a unique structure that is called Husserl's corpuscle. Now, what is this Husserl's corpuscle? So these are a rounded ovoid structure which are present in the medulla, eosinophilic hyaline mass at the center, surrounded by type 6 epithelio-reticular cells, which are concentrically arranged. Okay, that is the structure of Husserl's corpuscles. Okay, now please keep that in mind. Husserl's corpuscles represent the degenerating thymocytes, and their number increases with age. तो जो थाइमोसाइट्स है वो डिजेनरेट हो जाता है, देन दे आर फॉर्मिंग दिस स्ट्रक्चर हसल्स कॉर्पसल. जितना ज़्यादा होगा हसल कॉर्पसल, दैट मीन्स द एज इज ऑल्सो मोर इन केस ऑफ थाइमस. Next, this type 6 epithelio-reticular cell, they also secrete interleukin 4 and interleukin 6, which helps in thymocyte maturation. Okay, so these are some important functions of the epithelio-reticular cells. Okay, along with that, it also produces hormones thymosin and thymopoietin. So there are 6 types of epithelio-reticular cells in the thymus. Okay, as you can see, 3 are in cortex and 3 are in medulla, having different functions. Okay, among them, if you see, this one 6 is very important because it is helps in formation of the Husserl's corpuscles. Okay, after that, if you see the type 4, it is located adjacent to type 3. Along with type 3, it is forming the corticomedullary junction. Okay, and then type 5, cellular framework of the medulla. Okay, now this one type 1, what they do, they isolate the lymphocytes from connective tissue capsule, trabeculum and blood vessels. Okay, now these are also helping in the formation of the blood-thymic barrier. Okay, so 6 different types of epithelio-reticular cells along with the function. Next is the blood-thymic barrier. So in the beginning I have mentioned that it is a primary lymphoid organ. That's why barrier will be there. Barrier will protect it from getting exposed to the antigen. Now how the barrier is formed? So blood-thymic barrier has the following components. So always keep that in mind. Whenever you are discussing or writing about the barrier, you have to start from inside and then you have to go to the outside. ठीक है? Inside में क्या होगा? Blood vessel होगा. Okay, blood-thymic barrier, blood-brain barrier. Okay. So like this, whenever any barrier is there, how you have to describe it? Inside to outside. Inside में क्या रहेगा? Blood vessel रहेगा. Okay. तो first you can see capillary blood vessel, मतलब capillaries are there. So capillary endothelium and basal lamina is there. And along with that, few pericytes. These are specialized cells which are present, which controls the lumen of the blood vessel. Next, perivascular connective tissue with few macrophages. That is the second layer. Next, type 1 epithelio-reticular cells are there, which are of the which part? Cortex of the thymus. Okay. Then cells of the capillary endothelium and type 1 epithelial cells shows occluding junction. Okay. And now why this is present? It isolates T cells from circulating blood and antigen in it. That's why blood-thymic barrier is present. Here is the diagram. Okay. So here also you can see you have to go from inside to outside. So inside में क्या रहेगा? Blood vessel. Okay, तो blood vessel में here we are taking the blood capillary. So once you take the blood capillary, that means you are getting the capillary endothelium. See, this is the endothelium. Then this is the basal lamina. Okay, and then this is the pericyte. After that, perivascular connective tissue is there. As you can see, this is the perivascular connective tissue. Okay, then this is the basal lamina, and after that you can see type 1 epitheloreticular cell. Now here, from here, the thymus starts, the cortex of the thymus. Okay, and here you can see the T lymphocytes. Okay, so because of this barrier only, T lymphocytes cannot go and reach the blood vessel. Now this is the microscopic structure, both low magnification and high magnification image of thymus. ठीक है, तो इसमें क्या दिखाई दे रहा है? What you can see over here. So first of all, you can see the cortex. So start from here. See, this is the cortex. This is the trabeculae. ठीक है. Now this trabeculae is incompletely separating it into lobules. So this is the cortex. This is the medulla. ठीक है. This is the cortex. This is the medulla. Now ये देखो, ये एक lobule है, इसका medulla. ये एक lobule है, इसका medulla. You can see the medulla are continuous with each other. ठीक है. Now if you concentrate on the medulla, you can see the Husserl's corpuscle. ये देखो, ये pink color का structure, dark pink color का structure दिखाई देगा, dot dot. In case of medulla, these are the Husserl's corpuscle. Now if you zoom it, so this is under low magnification. If you zoom it under high magnification, then the appearance will be like this. Okay, so this is how the Husserl corpuscles look like. So very easy to identify the thymus. First of all, you will see the cortex, which is darkly stained. Then if you come towards the medulla, you can see it is lightly stained. Next, in the medulla, if you concentrate, you will be getting this dark pink color structure. This is nothing but the Husserl's corpuscle. Once you zoom it, you can see the Husserl corpuscle easily. Along with that, you can see the trabeculae, which are incompletely dividing the lobules. Okay, so that is all about the thymus. Now some other structures are also left. For example, tonsil is there. Okay, then mucosa associated MALT, lymphoid tissue. Okay, so those things we'll discuss once we start that particular part. Okay, for example, MALT will discuss in respiratory system. Okay, GALT is there, gut associated lymphoid tissue, that will discuss in intestine part. Okay.